New Zealand, a fascinating island in the Pacific Ocean. Known for its outstanding diverse nature and wonderful wildlife. From its captivating volcanic areas, serene and secluded beaches, to its boundless and breathtaking landscapes. Along with its crystal clear rivers. This island presents a natural wonderland. However, lesser-known creatures also inhabit this island, and they are no less captivating. These animals, only 1 centimeter in size and well camouflaged, inhabit the river systems of the North Island, the freshwater snail, Latia neritoides. As defense, it releases a sticky mucus. 2 sites are supposed for its protection: the pneumostome area or the lateral surface of the foot. Now it might be worth mentioning that the mucus of Latia close. We are following the mantle cavity roughly to the middle of the animal. Ultrastructural investigations as well as histochemical stainings should provide more information about the present gland cells. 2 gland cell types could be identified, which are possibly involved in the glowing mucus production. The TM image shows the release of the granular content. The staining results of this type are comparable to the fluorescent mucus. And then there is the second one, maybe a whole green gland cell type, appearing as often as the first one, with no staining reaction. 2 big gland types are dominating the lateral foot area. Again, histochemical stainings were made to determine if proteins or carbohydrates are present. L2L doesn't react to any of them. But lectin affinity tests show carbohydrates are present, and EDX measurements reveal a relatively high sulfur concentration. The content of this gland cell type is fine granulated. In both L1L and the defensive mucus, carbohydrates and proteins are identified. The results of the lectin affinity tests differ in contrast to L2L. Magnesium and phosphorus are found in elevated concentration. This gland cell type is stuffed with vesicles that disrupt when released. Both gland cell types seem related to the defense system, primarily responsible for mucus viscosity. No gland-specific muscles are detectable. The gland cells are embedded into the palatal muscle network and their content is released by contractions of the foot. Diatoms are found on the shell of these snails. We are following the digestive tract, starting at the esophagus, followed by the gizzard, and then the midgut gland appears. Underneath the esophagus, the radula with musculature becomes visible. By looking deeper inside the salivary gland, we can see that only some of the cells contain carbohydrates. The cells of the midgut gland, in contrast, respond less strongly. Investigating the intestinal loop, two gland types with an uneven distribution pattern and different staining characteristics are noticeable. Appearing in fainted blue, we can see the reproductive tract of Latia. Once again, specific gland cell types were the suspect of investigation, with the most intriguing ones being found in the region of the sperm oviduct, colored in orange. These are the only ones that reacted positive for basic proteins. Parts of the nervous system and the excretion organ become discernible, and it is known that every animal needs a heart.